Hi everybody, welcome back to chapter 3. We're working on structures that are found within prokaryotic organisms now. And so when you look at a prokaryotic cell, they look very different from eukaryotes because they don't have all the organelles that we see in eukaryotic organisms. So prokaryotes have a region called a nucleoid where it's just a region within the cytoplasm where all the DNA kind of aggregates together, okay? They don't have organelles in general. They do have ribosomes, which are considered organelles. So in that sense, they have at least one type of organelle, but in general, they don't have those typical complex organelles that eukaryotic cells have. They can be different sizes and they can be different shapes, but they're always single-celled organisms. There we go. So when you look at the structure of prokaryotic cells, bacterial cells and archaeal cells are always going to have certain types of structures that are super common, like they can be coccus, like this. They can be rod-shaped or bacillus. So coccus is round and circular. Bacillus is rod-shaped and, well, rod. Vibrio is a rod that is curved. When you look under a microscope, it's very hard to distinguish between a rod and a vibrio. So you really don't see the differences here. You can have some cells that are called coccobacilli, so they look more like large ovals. Less common, but still structures that we see are spirillum. Spirillum are spiral-shaped, and then spirochetes are long and helical spirals. So this is more like a loose spiral, whereas this looks more like that helix that you see in the DNA. The most common of these shapes is coccus and bacillus. But even with coccus and bacillus, we can have different groupings or arrangements. So I told you already that prokaryotic cells are always single-celled. They're always, you never have a multicellular prokaryote. They're single-celled, but they can grow in arrangements where you have multiple single cells that cluster together. And so if you have a single coccus cell, it's just known as a coccus. But if you have two coccus cells that connect, when they replicate, they stay connected. This would be a diplococcus or diplococci. If you have four coccus cells that stick together, they form like a square-like structure. This is called a tetrad. If you have more than two coccus that are sticking together, this is known as streptococcus or streptococci. If you have a cluster of coccus cells, kind of looks like a group of grapes that are sticking together, this is known as staphylococcus or staphylococci. And then for bacillus, we also have single rod would be just a single bacillus. If you have more than one rod and they are forming a chain, this is known as streptobacilli or streptobacillus. So here you can see again that nucleoid region where DNA is going to be found in the prokaryotic organism. It's just a region inside. There's nothing separating the DNA from other structures in the cytoplasm of the cell. The DNA is in this nucleoid region, and then ribosomes, which build proteins from the DNA, sit right outside the nucleoid region. And so they are known as 70S ribosomes. 70S ribosomes are composed of two different subunits. So the subunits are known as 50S subunits and 30S subunits, but together they're called 70S ribosomes. Those differ in structure from eukaryotic cells. And so because of this, we can actually target protein synthesis in bacteria when we're trying to get rid of an infection. Ribosomes also differ in structure between archaea and bacteria. So we can identify archaeans and bacteria by their ribosomes. So I told you that they have a different size. I told you they had a different structure than eukaryotic ribosomes. They are 70S ribosomes. So here you have the large subunit. This is a 50S ribosome. Here's the small subunit. This is a 30S ribosome, but when they come together, they are called a 70S ribosome. The S stands for a Svedberg, an individual who identified these different sizes. And so they're just known as Svedberg units, okay? And so we can separate organisms based on their ribosomal structure and size because they differ between eukaryotes and bacteria as well as archaea and bacteria. Prokaryotic cells also have inclusions. Inclusions are storage compartments for nutrients or chemicals. They can store gases. They can also store waste products. And so over here you can see different substances being stored, polyhydroxybutyrate lipid droplets and inclusion bodies. Where's another one? Where's it? D. These contain magnetosomes, which allow the cells to actually be pulled by the different polar caps, the magnets in the polar caps. And so they can actually, or those magnets actually are, those magnetosomes are magnetic. That's what I wanted to say. And then bacteria also have structures known as endospores. So we talked a little bit about endospores when I was talking about the endospore stain. The endospores, and when we were talking about spontaneous generation and debunking it. Endospores are structures formed when survival is unfavorable for a cell. So certain types of cells can produce endospores if conditions are not favorable for growth. And when this happens and they produce endospores, then they can, like, I won't say live, they can be or stay in the environment until the environment becomes favorable. They can exist for days, weeks, months, even years in an environment, and once the environment becomes favorable again, the cell can germinate. So basically come back. This is not a form of replication because it's the same cell that forms the endospore is what is going to be produced. So it's kind of like a hibernation for the cell in a sense. And so for endospores to form, the environment has to be unfavorable. So you have the DNA starts to replicate. Once the DNA replicates, you have two sections of DNA, identical DNA. One moves into what we call a spore area. The other DNA, this is the mother spore, okay? The spore forms another membrane around itself, and then a protective cortex forms around that. There is the structure, the endospore is then dehydrated, so water is pulled out, and then the endospore is released from the parent cell. The parent cell dies, and that endospore with the existing DNA is released and can sit in the environment until time for, or until favorable conditions come again. These you can see down here, these are endospores, these little green structures. The cells that are producing them, obviously you can see them as well. They are pink. And so this is an example of Bacillus cereus producing endospores. I always, when we do the endospore stain, I always have, or I try to have, a new Bacillus cereus and old Bacillus cereus. The reason I do that is because our Bacillus cereus, if it's brand new, if it's, you know, within 24 hours, it doesn't produce the number of endospores that you see if the Bacillus cereus are old. Because if it's old, then they're going to produce a lot more endospores because the environment is no longer favorable. Does that make sense? Or I shouldn't say that because it should make sense, but if it does not make sense, then you can ask me. And here's another picture of an endospore from your textbook. All right, the last thing I'll talk about, because this is 11 minutes already, is actually, you know what? I'm going to stop here because the plasma membrane is a pretty complex structure. So we'll stop here and we'll get into the plasma membrane in our next video. Okay, bye.